Welcome back, everyone, to the deep dive. This is where we really get into the weeds, pulling apart complex topics to, find those nuggets of information that help you make sense of the world. Today, we're heading into a really interesting, and I think for many people, quite a personal area. Complementary and alternative medicine, or CAMN, specifically how it relates to cancer care. And our focus is going to be on a pretty well-known herbal blend called Essiac Tea. Maybe you've heard of it.
It's definitely got a presence.
Right.
Yeah.
But before we dive into Essiac itself, I think it's important to set the stage a bit, talk about prostate cancer, because that's really the context for this study we're looking at today. It's a huge health issue, isn't it?
It absolutely is.
Yeah.
Our sources make that very clear. For American men over 50, it's actually the second most common cancer diagnosis.
Second most common. Wow.
And sadly, it's also the second leading cause of cancer death for men in the U.S. And if you look globally, it's the fourth most commonly diagnosed cancer in men. So it's a worldwide concern.
That really puts it in perspective.
Yeah.
And there are variations too, right? Like who gets it most often?
Yeah, the data shows incidence rates are highest in African Americans and lowest in Asians. And age is a massive factor. It increases really rapidly as men get older.
How rapidly?
Well, autopsy studies are quite revealing. They show that by the time men reach age 75, somewhere between 50 and 70%, that's, more than half show at least one focus of prostate carcinoma.
50 to 70%. That's staggering. So it's incredibly widespread.
Exactly. And when you're facing a diagnosis like that, something so common and potentially serious, it makes perfect sense that people explore every single option available to them, which brings us right back to CAM.
The complementary and alternative approaches.
Right. It's a really noticeable trend among cancer patients generally. People often combine their standard medical treatments, chemotherapy, radiation, surgery with other things. You see vitamins, minerals, herbal medicines, green tea, maybe special diets.
And Essiac sits right in there.
It really does. Our sources pointed out just how popular it is. In one survey of cancer patients looking at CAM use, Essiac came in as the fifth most commonly tried treatment.
Fifth. That's high up the list.
It is. Nearly 15%, 14.8% to be precise, of the patients surveyed said they'd used Essiac. For an herbal tea operating largely outside conventional medicine for so long, that's pretty significant. It definitely makes you ask, why? What's the draw?
Absolutely. So let's get specific. What is Essiac? What's in this blend?
Okay, so Essiac is a specific mix of 4 herbs. You've got burdock root, the scientific name is Arctium lapa. Then there's Indian rhubarb root, which is Rheuma officinale, slippery elm bark, that's Ilmus fulva, and finally sheep sorrel, Rumex aceticella. Just those 4.
And where did this recipe come from? It has a bit of a story, doesn't it?
It does, yeah. The origins are reportedly traced back to Ojibwa and Native American medicine men, but it was really brought into the spotlight by a Canadian nurse named Rene Caisse.
Rene Caisse. Essiac.
Yeah.
I see it now. Her name's spelled backward.
Exactly. Pretty clever naming. She actually ran a cancer clinic up in Bracebridge, Canada for many years, giving the tea to patients. And her clinic, well, it relied heavily on anecdotal evidence, stories of success.
And those stories fueled its marketing, right? Our sources mentioned enormous amounts of testimonial evidence.
For sure. For over 20 years, it was marketed with these powerful testimonials. People claimed it improved their quality of life, helped them live longer. Some even suggested it cured their cancer.
Which are incredibly hopeful claims for someone dealing with cancer.
Absolutely. But, and this is the crucial point leading into our deep dive, despite all that anecdotal weight, none of those claims about cures had actually been scientifically proven.
Right.
And maybe even more surprising, when the key study we're discussing was published back in 2004, there were essentially no published scientific studies looking at the combined Essiac formula. Just the individual herbs maybe, but not the whole package as people were actually using it.
So a huge gap between belief and scientific evidence.
A really significant gap.
Okay, so that's where this 2004 study comes in. Inhibition of prostate cancer cell proliferation by Essiac, by Ottenweiler and colleagues. They were trying to bridge that gap, weren't they? Look at the combined effect.
Precisely. Their goal was to finally investigate the combined blend, see if it actually inhibited cancer cell growth, and importantly, start to understand how it might work. Looking at those potential mechanisms of action, including effects on the immune system.
Okay, let's unpack this study then. Let's dig into what they found. So we know there was this lack of science on the full Essiac blend, even though people were using it. But the researchers weren't starting completely blind, were they? They had some clues from the individual herbs.
That's right. Before this particular study, there was already some groundwork laid. The University of Texas Center for Alternative Medicine Research, UTCAM, had done a big literature review.
Okay.
They looked at 107 different publications about the four herbs in Essiac. And what they found was pretty telling. About 68% of those papers were directly related to cancer in some way.
So almost 70% focused on cancer aspects of these individual herbs.
Yeah. And they also confirmed what we just talked about. Lots of testimonials for the blend, but crucially, no clinical trials on the combined Essiac itself.
Right.
But, and this is key for understanding the potential mechanisms, they did find studies on the individual herbs. Specifically, 13 studies done in vivo, so in living animals, and 11 studies done in vitro, meaning in lab dishes. And these looked at how each herb on its own affected cancer. That provided the initial hints, the foundation for figuring out how Essiac might work.
Okay, so let's get into those individual herbs, because this is where we start seeing the potential mechanisms, the biological actions. Let's start with burdock root, Arctium lapa. It sounds like that one got a lot of attention.
It really did. Burdock root is, well, it's pretty packed with beneficial compounds. For starters, it contains several potent antioxidants, things like flavonoids and polyphenols.
And antioxidants are basically protective for our cells,
Right.
Fighting off damage.
Exactly. They neutralize those unstable molecules, free radicals, that can damage DNA and contribute to diseases like cancer. So that's one mechanism right there, basic cellular protection.
Okay, what else?
Well, even more specifically, back in 1984, researchers identified something called a desmutagenic factor in burdock root extracts.
Desmutagenic. Okay, break that down for us.
Right. It sounds complex, but it's actually a really important concept. Mutagenic means something causes mutations, changes in DNA. So desmutagenic means it reduces or prevents those mutations. This factor in burdock root was found to lessen the DNA damage in cells that were exposed to toxic chemicals.
So it's helping prevent the initial damage that could even lead to cancer.
Precisely. It interferes with the ability of harmful chemicals to cause mutations. And interestingly, this factor was pretty robust. It could withstand heat and digestive enzymes, suggesting it might actually, survive digestion and still be active. That points to a really fundamental protective mechanism.
That is fundamental. Are there specific chemicals in burdock identified for these effects?
Yes. One mention is quercetin pentamethyl ether. It's a type of flavone, a plant chemical. Studies showed it inhibited lung tumor formation in mice by about 50%.
50% inhibition, that's substantial.
It is. But maybe even more relevant, mechanistically speaking, are compounds called isoflavones found in burdock root, specifically biochanin A and genistein.
Isoflavones, I've heard of those, often linked to soy, right?
Yeah.
But they're in burdock too.
They are, and these particular ones showed really strong effects in lab studies. One study by Yanagihara and colleagues in '93 found they strongly inhibited the proliferation of 3 different types of human stomach cancer cells.
Stomach cancer cells.
And they moderately inhibited other types too, like esophageal and colon cancer cell lines, so pretty broad action against different digestive system cancers in the lab.
And how were they inhibiting them? Was it just slowing growth or something more dramatic? This gets to the mechanism, right?
Exactly. This is where it gets really interesting at the cellular level. The researchers observed that when they treated the cancer cells with these isoflavones, they saw chromatin condensation and nuclear fragmentation.
Okay, what does that mean in simple terms?
Think of the cell's control center, the nucleus containing the DNA. Normally it's all organized. Chromatin condensation and fragmentation mean that the DNA is clumping up and the nucleus is breaking apart. These are classic signs of a cell undergoing programmed cell death, or apoptosis.
Ah, apoptosis, the cell self-destruct mechanism.
Precisely. And they found this apoptosis was dose-dependent, more isoflavone, more cell death. This is huge because cancer cells are often defined by their ability to avoid apoptosis. They refuse to die when they should.
So these compounds were basically triggering the self-destruct sequence in the cancer cells.
That's a great way to put it. The study concluded that these isoflavones worked by activating the specific signaling pathways for apoptosis within the cancer cells. They were essentially sending the self-destruct signal. And importantly, this wasn't just in a dish strip. Follow-up studies showed these compounds could suppress tumor growth in mice too. That really strengthened the case for them being potential anti-cancer agents.
Wow. So activating apoptosis is a major mechanism there. Anything else for burdock?
Yes. There's also evidence for anti-inflammatory effects. Another study showed burdock extract reduced inflammation in rats and also protected the liver from chemical damage. The researchers linked this back to its antioxidant and radical scavenging abilities.
Which is important because inflammation is linked to cancer development too, right?
Absolutely. Chronic inflammation can create an environment that helps cancer grow. So, by tackling inflammation and oxidative stress, burdock seems to be working on multiple fronts, not just directly telling cancer cells to die.
Okay, burdock root is clearly a heavy hitter in this blend. What about the others? Slippery elm bark, Ulmus fulva. What does it contribute?
Slippery elm, based on the literature review, is primarily noted for having anti-tumor, anti-inflammatory, and antioxidant activities.
So, similar properties in a way.
Yes, contributing similar types of potentially beneficial actions. The term anti-tumor is broad, but it suggests compounds that can interfere with tumor growth or survival. Adding anti-inflammatory and antioxidant effects just strengthens that profile. It likely adds another layer of complementary actions to the blend.
Got it. And sheep sorrel, Rumex acetosella.
Sheep sorrel is interesting because it's also cited for anti-inflammatory, antioxidant, and anti-cancer properties. Plus, it seems to have some antibacterial activity as well.
Okay, so another multitasker.
Seems like it. Again, the anti-cancer part suggests compounds that might inhibit proliferation or maybe promote cell death, adding to the overall potential synergy of the Essiac formula.
And the final one, Indian rhubarb root, Rheum officinale.
Right. The key compounds highlighted there are anthraquinones. Now, these are maybe best known for their laxative effects in other contexts, but studies have also shown they have experimental anti-inflammatory, antiseptic, antispasmodic, and crucially for us, anti-tumor activities.
Anti-tumor again, so suggesting direct effects against cancer cells.
That's the implication. These compounds might disrupt cancer cell division or trigger death pathways, similar perhaps to some conventional drugs, but obviously derived from a plant source.
So when you look at all four together, burdock, with its apoptosis, triggers, and antioxidant power, slippery elm and sheep sorrel adding anti-tumor and anti-inflammatory effects, and Indian rhubarb with its anthraquinones. It starts to paint a picture of potential synergy.
Exactly. You have multiple herbs, each with compounds showing anti-cancer, anti-inflammatory, antioxidant properties in individual studies. It builds a strong rationale for why the combined blend might be effective.
But, as we said, nobody had really tested that combined effect scientifically. Which brings us squarely back to the Ottenweller study. Here's where it gets really interesting, seeing if the whole was more than the sum of its parts. Okay, so let's dive into the specifics of that Ottenweller's study design. They needed to test the combined Essiac blend. What cells did they actually use?
Right. They chose their cell lines carefully. First, they used Rene Caisse cells. These are human prostate cancer cells actually isolated from a lymph node metastasis of a patient. So highly relevant for prostate cancer research.
Okay, real human cancer cells. What else?
They also used CHO cells, Chinese hamster ovary cells. Now, these aren't cancerous, but they are considered transformed. That just means they have genetic changes that let them grow indefinitely in the lab. They served as a sort of non-cancerous comparison point.
So comparing effects on cancer cells versus non-cancers but still altered cells makes sense.
And they maintain these cells very carefully. Standard lab conditions, temperature, nutrients, CO2 levels, to make sure the results were consistent and reliable. Good science practice.
And the Essiac itself, how did they prepare it to make sure they were testing what people actually use?
That was critical. They bought the commercial Essiac powder from Essiac Canada International, just like someone would from a health food store, and they followed the package directions precisely for brewing it.
Boiling, cooling, simmering. The whole process.
Exactly. They measured the powder, added water, boiled it for 10 minutes, let it cool for 4 hours, then simmered it again for 5 minutes. And they always made it fresh, within 24 hours of using it in the experiments.
Okay. And they adjusted the pH. Why was that important?
Very important. They adjusted the Essiac brew to pH 7.2, which is the same pH as the growth medium the cells live in. If the Essiac made the medium too acidic or too alkaline, that pH change itself could affect cell growth. They needed to isolate the effect of the Essiac components, not just a pH shift.
Right, controlling the variables.
Exactly. And then they filter sterilized it to remove any potential bacteria and used plain filtered water in growth media as their control group for comparison. Really thorough preparation.
Definitely sounds rigorous. So they have the cells. They have the carefully prepared Essiac.
Yeah.
How do they measure if it was actually stopping the cells from growing? What was the method?
They used a standard technique called a cell proliferation assay. Basically, they put a known number of cells, 100,000, into little wells in a plate. After letting the cells settle for 24 hours, they added different amounts of the Essiac preparation to some wells and just the control solution to others.
Okay.
Then, to measure growth, they added something called tritiated thymidine. It's a building block for DNA, but it's radioactive, so it's traceable.
Ah, the growth marker you mentioned.
Right. Cells only take up thymidine when they're actively making new DNA to divide. So after letting the cells incubate with the Essiac and the marker for a couple of days, they measured how much radioactivity was incorporated into the cell's DNA.
More radioactivity means more DNA synthesis, which means more cell division and growth.
Exactly. Less radioactivity in the Essiac-treated wells compared to the control wells meant the Essiac was inhibiting proliferation. And they did this multiple times, at least 3 runs for each assay to be sure of the results.
Okay, that covers the cancer and transformed cells, but they also looked at immune cells, right? How did that work?
Yes, that was a really important part of the study. They took spleen cells from healthy mice. The spleen is full of immune cells, especially T lymphocytes.
The T cells that fight infection and cancer.
Correct. They stimulated these spleen cells with something called concanavalin A. It's a substance that basically jump-starts T cell proliferation, kind of mimicking an immune response activation.
Okay.
Then they treated these activated immune cells with different doses of Essiac, or the control solution, and used the same radioactive thymidine method to measure how much the T cells were proliferating. This let them see if Essiac helped, hindered, or had no effect on this key immune cell activity.
And do they just rely on the radioactivity counts, or do they also, look at the cells?
Good question. They also did regular microscopic observations. They visually checked the cells throughout the experiments to see if there were any obvious changes in their shape or appearance, their morphology. Were the cells dying, looking stressed, or just quietly dividing less?
So combining the quantitative measure with qualitative observation.
Exactly. And for the final analysis, they crunched the numbers from the radioactivity counts, calculated the percentage of inhibition compared to the controls, ran the experiments multiple times, at least 6 times each, and used statistical tests to determine if the effects were significant. Really solid methodology.
It certainly sounds like it. Okay, methodology covered. Let's get to the payoff. What did they actually find? What were the results, starting with those non-cancerous CHO cells?
The results were pretty striking. For the CHO cells, the non-cancerous transformed line, they saw clear dose-dependent inhibition.
Meaning more Essiac led to less growth.
Precisely. Even a small amount, 5 microliters, inhibited proliferation by 53%. And when they increased the dose to 35 microliters, the inhibition jumped to 80%. So a strong effect even on these non-cancerous altered cells.
Well, no visible damage.
Interestingly, no. They reported no major morphological differences. The cells weren't obviously dying or falling apart, they were just growing less. Suggests something more subtle than just killing them outright.
Okay, that's interesting in itself. But the big question, what about the actual human prostate cancer cells, the LNTAC line?
That's where the results were arguably even more significant. They saw the same powerful dose-dependent inhibition with the LNCAP cells, but, and this is the really exciting part, the percentage of inhibition was higher for the LNTAP cancer cells than it was for the non-cancerous CHO cells at the same doses.
Higher inhibition for the cancer cells, so it was working better against them.
Exactly. It suggests a degree of selectivity. For example, that 5 microliter dose that inhibited CHO cells by 53%, it inhibited the LNTAP cancer cells by 71%. And the higher 35 microliter dose pushed inhibition in LNTAP cells to about 91%.
91% inhibition of prostate cancer cell growth. That's huge.
It's a very strong in vitro effect. And again, like the CHO cells, they didn't observe major morphological changes, pointing towards an inhibition of the growth process itself rather than just cell destruction.
That selectivity is really key, isn't it? Finding things that hit cancer harder than normal cells.
It's incredibly important. That's often the goal in cancer therapy development.
Okay, so direct inhibition, potentially selective for cancer cells. Now, what about the immune side, the spleen cells? Those results sounded particularly nuanced.
They were definitely nuanced and really fascinating. Remember, they were stimulating these immune T cells to proliferate, then adding Essiac. What they found was that the effect was completely dependent on the dose.
How so?
At low doses of Essiac, they actually saw enhanced proliferation of the spleen cells. The immune cells grew more.
So at low doses, it seemed to stimulate the immune response.
That's what the data suggests, yes, an immune-boosting effect. But And here's the twist: at the higher doses, the same concentrations that were strongly inhibiting the CHO-LNKP cells.
Yes.
At those higher doses, Essiac became inhibitory to the spleen cell proliferation too. It started to suppress the immune cell growth.
Wow. So boost at low doses, suppress at high doses. That's complex.
It is, and the really critical comparison they made was this. The very same low concentrations of Essiac that were stimulating the immune spleen cells were already significantly suppressing the growth of both the CHO and LNKP cells by at least 45%.
Okay, hang on. So at a dose where it's boosting immune cells, it's already significantly inhibiting the cancer cells.
Correct. That's the crucial takeaway. It points towards this potential sweet spot, a therapeutic window where Essiac might simultaneously help activate the immune system and directly put the brakes on cancer cell growth, a very specific differential effect.
Okay, this dual effect is really where things get interesting.
Yeah.
This raises an important question. What does this actually tell us about how Essiac might be working? The mechanism behind it all.
You nailed it. This is where we synthesize those results. We clearly have the in vitro evidence. Essiac slows down the growth, the proliferation of both transformed cells and, more potently, cancerous prostate cells. That's direct inhibition.
Right.
But then you layer on this variable immune effect enhancement at low doses, suppression at high doses. That adds a whole other dimension.
Which leads to this dual-edged sword idea you mentioned.
Exactly. That's what the researchers propose, and the data supports it. Think about it. At an optimal concentration, potentially a lower dose, Essiac might stimulate those cytotoxic T cells.
The killer T cells that target cancer.
Right. So part one of the mechanism could be augmenting the body's own immune attack against the tumor, helping the immune system do its job better. An indirect but potentially very powerful anti-cancer effect.
Okay, that's one edge of the sword. What's the other?
The other edge is the direct inhibition we saw. Simultaneously, Essiac could also be directly inhibiting tumor cell growth, stopping those LNCAP cells from multiplying, independent of the immune system.
So it's attacking from two angles, boosting the defenders and slowing down the invaders.
That's a great analogy. And that's why they called it potentially ideal for combating tumor cell growth. It's a multi-pronged approach. And the potential immune boost could be especially relevant for patients whose immune systems might be weakened, say, by conventional treatments. It suggests Essiac might work with the body.
That makes a lot of sense. And you mentioned the T cells producing things like IL-2 and interferon gamma when activated. Can you elaborate a bit on why that specific immune activity is helpful against cancer?
Sure. So when T cells get activated, like maybe by low-dose Essiac, they ramp up production of these signaling molecules called cytokines. Interleukin-2 or IL-2 is like fuel for T cells. It helps them multiply rapidly, building up an army of cancer-fighting cells.
Okay, so more troops.
Exactly. And interferon gamma, or IFNG, is another powerful one. It can directly slow down cancer cell growth, but it also acts like an alarm signal, making cancer cells more visible to the immune system and recruiting other immune cells to the fight.
So it's not just more troops, but better coordination and making the enemy easier to spot.
Precisely. So seeing evidence of T cell activation, even indirectly, through increased proliferation in response to Essiac, suggests that the phytochemicals in the T could potentially be triggering these beneficial downstream immune effects. It's interacting with these complex communication networks within our immune system.
It really paints a much more sophisticated picture than just herbal tea shrinks tumors. It's about modulating complex biological systems. So connecting this back to the bigger picture, these are exciting in vitro findings, but what's next? What more do we need to know to really understand Essiac's potential?
That's the crucial question. These lab results are a fantastic starting point, but they open the door to many more necessary investigations. The researchers themselves outline the key next steps. First off, identifying the active components.
Figuring out which chemicals in the blend are doing the heavy lifting.
Exactly. Which specific phytochemicals are responsible for inhibiting the cancer cells? Which ones are modulating the immune cells? Is it one compound or a combination? Answering that would allow for much more targeted research, maybe even standardization of the product. It gets us closer to understanding the precise molecular mechanisms.
Makes sense. What else? Moving beyond the Petri dish.
Absolutely critical. In vivo studies are essential. That means testing Essiac in living animals, like mice. You could give them Essiac, maybe in their water, and then see how their immune system responds to challenges like exposure to a virus or, more relevantly, the growth of implanted tumors.
To see if the effects hold up in a whole complex organism.
Right, because the body metabolizes things, distributes them differently. What happens in a dish doesn't always translate directly. In vivo studies start to bridge that gap.
And speaking of metabolism, you mentioned the liver earlier. How can research account for how our bodies process these compounds?
That's another key suggestion. Trying to mimic the body's environment more closely. For instance, before testing Essiac on cells in a dish, you could pre-treat it with liver microsomes.
Those enzyme-rich liver cell fragments.
Yes. That simulates what happens when compounds pass through the liver. Does the liver break down the active components, or does it maybe activate them, turn them into something more potent? Understanding that metabolic fate is vital for predicting real-world effects in humans.
So active compounds, animal studies, metabolic studies. What's the ultimate goal?
Human clinical trials. Given that Essiac is already widely used, readily available as an herbal tea, the researchers pointed out that setting up trials to rigorously evaluate its effects in actual cancer patients would be relatively straightforward.
The gold standard.
The gold standard. Does it improve outcomes? Does it affect quality of life? Are there side effects? What's the optimal dose? These initial in vitro results showing selectivity and immune effects absolutely warrant such a study, as the researchers put it. That's how you move from promising lab data to something that can reliably inform clinical practice and patient choices.
It's quite a path from traditional use to potential clinical validation. So what an exploration that was. We started with Essiac Tea as this popular, almost folkloric remedy surrounded by testimonials.
Lots of stories.
And we've ended up looking at specific molecular mechanisms inhibiting prostate cancer cell growth, potentially more than non-cancerous cells, and this really fascinating dual effect on the immune system. This Ottenwiller study didn't provide the final answer, of course, but it gave us some real, tangible scientific insights into how it might work.
It really did. It moved the conversation forward, providing that crucial preliminary data. And for me, what's truly fascinating here is this ongoing quest to understand compounds from nature. You know, how do these plant chemicals actually interact with our incredibly complex biology?
Right.
And how even remedies that have been around for ages, steeped in tradition, can reveal these really sophisticated mechanisms when we finally apply modern scientific methods. It just makes you wonder, doesn't it? What other secrets are out there in nature just waiting for us to look closely enough?
That is a great thought to end on. It definitely sparks curiosity. And on that note, that wraps up our deep dive for today. Thanks so much for joining us on this journey into Essiac Tea and the science behind it. Keep asking questions, keep exploring. We'll see you next time on The Deep Dive.